Hey SFS listeners, Connor Wonder here. I'm super excited to bring you the next installment of the Alzheimer's series that we've been putting on. This one in particular is awesome because it was a true Alzheimer's disease roundtable. So it's put on by myself. I was hosting it and kind of directing some questions. But really, it's three excellent scientists working on the forefront of Alzheimer's disease research and knowledge, pushing the boundaries as we know them and as we begin to understand them more. And they're all talking back and forth. We actually had one mic going around a table. It was like a bar style table. Everyone was standing up. This is after a great dinner put on by BrightFocus, which is of course the host of the conference. And it just, you get to know the scientists, you get to know the science, and you get to know Alzheimer's disease like I've really never heard it communicated. So super excited to bring this to you all. I know the audio quality may not be perfect. I'm actually pretty impressed at how this works. It's the first time I've done it, but it's amazing. I don't want to give too much away. You are going to learn a lot, and it's just a fun listen. One last thing before we get started, I really want to thank Rachel and Martha from the BrightFocus Foundation for helping me organize all of these podcast interviews, whether they were during a coffee break or after dinner or at random times all throughout the conference event. I couldn't have done it without them, and they helped me organize the show notes as well. So huge shout out to them, and of course the BrightFocus Foundation and everyone involved in putting on that conference. On this episode, in order, we're going to have Dr. Cynthia Lemere, Dr. Charles Glabe, and Dr. Leah Grinberg. Enjoy. So if you all could introduce yourself briefly and what you work on in kind of simple terms, and then we'll break the science down later. First off, thank you all for agreeing to do this. I really appreciate it.
Thank you, Connor. My name's Cynthia Lemere. I'm a scientist at Brigham and Women's Hospital and an associate professor of neurology at Harvard Medical School in Boston, Massachusetts. And I've been working in the field of Alzheimer's for about 30 years now. I actually started in Alzheimer's at UCSD as a technician and then moved to Boston and did my PhD there and studying Alzheimer's disease and Down syndrome. And since that time, I've been looking at different animal models for Alzheimer's disease and trying to understand better the mechanisms underlying the changes in Alzheimer's disease, how Alzheimer's disease occurs, why it's in specific parts of the brain, and also trying to find therapies to prevent Alzheimer's disease mostly. And I'll turn it over to my colleague, Dr. Charlie Glabe.
Hi, my name is Charles Glabe. I'm a professor at UC Irvine, and I've been working on Alzheimer's disease a little less, probably about 28 years. And I'm interested in several things. I'm interested in the structure of amyloid because the structure of a biomolecule is usually related to its function, and it's possible that amyloid might be doing something bad in the disease. A lot of people think it does. I'm also interested in the mechanism of pathogenesis, why things go bad, why people get demented, particularly about how neurons die in Alzheimer's disease. So one of the things that's known is neurons die, and people argue about how they die, why they die, and the relationship of neuronal death to the other types of pathology like amyloid A beta and tau pathology. And lastly, I think we're all interested in how can we make things better? How can we prevent the disease if we can't cure it? And I'm interested in immunotherapeutics.
All right, so now it's my turn, and my name is Leah Grinberg. I am a associate professor in neurology at the University of at UCSF, University of California in San Francisco. I'm originally from Brazil, so you see my English is not perfect as for my colleagues, but I think we'll have to live with it. So I'm a neuropathologist, and for those that don't know what the neuropathology does, I am the person who is always late but always right. So I have the privilege to examine tissue from people who passed already, but they donate the brain when they were living for research. And what I can see is that in many people that they don't have any memory loss or cognitive decline or anything like that, they already may have changes in the brain, changes that we may call Alzheimer's disease or some other neurodegenerative disease. So it makes clear for me that, what we call normal aging, it's maybe a very elastic term. So with my research, what I try to bring is this knowledge that I can see in post-mortem tissue for living patients. Can we diagnose disease changes earlier than we do now? And if we can do it, can we delay the disease to show up, or can we even cure the disease? And my lab concentrate a lot in this kind of questions.
Well, thank you again. Those are great introductions. And I sense a common theme, which is, one, we need to start looking earlier in the disease. And that's something that we've been talking about basically the whole conference. We've been discussing kind of the shortcomings, the failures of previous clinical trials. And while we might be doing some of the right things, we aren't necessarily doing them at the right time. So we need to start moving ahead in stages. But Dr. Grinberg, you brought up a really good point, and something that I've been going back and forth on a long time, whether Alzheimer's disease is just accelerated aging or if it's some sort of special age-related disease. There's really something nefarious going on, something really terrible happening. And I was wondering if I could get all of your thoughts on that, because every person I ask in the field has kind of a different opinion on that matter.
Alzheimer's disease is very common, but it's not universal. There are many people who age and they don't get Alzheimer's disease. So I think the example that inspired me the most is how lung disease and heart disease was seen 50 years ago. So 50 years ago, if someone who was a little bit older and will go around spitting, people will say, you age, you accumulate water in your lungs, and this is okay, this is just normal aging. And now we know that there are some bacteria that cause it. And they are different bacteria that cause pneumonia in younger people. So we have to treat in a different way, but we treat it. And older people don't go around spitting because they don't have water in the lungs. The same is for heart disease. People don't drop dead because we have ways to detect atherosclerosis or high cholesterol, things that will lead to someone dropping dead if we don't do anything about it. So I feel that maybe for the brain, we can get to the same place. We will be able to understand what is really accelerating the process that will lead to dementia or Alzheimer's disease dementia, other dementia, and maybe be able to do something about it. And hopefully in 50 years, brain disease will be the same as lung disease and heart diseases nowadays.
This is Cindy, and I completely agree with you, Leah. And I also think that we've learned a lot from the genetic causes of Alzheimer's disease. So, for example, many years ago in my PhD work, we studied Alzheimer's disease in people with Down syndrome. And most people with Down syndrome, if not all people with Down syndrome, develop Alzheimer's pathology by approximately age 40. However, the age of onset of dementia varies tremendously. So there are some clues there that we need to figure out what is it that's accelerating the pathology and accelerating the dementia once somebody has pathology. It seems that some people can resist the dementia stage and live with the pathology, and that's true for people without Down syndrome as well. So there are people walking around over 65 years of age with a head full of amyloid that don't have signs of dementia. Whether they would go on to develop dementia within the next 10 years is still up for debate. And a lot of research shows that they probably could, or that they would be at least at high risk for that. So I think one of the key areas right now is really understanding better what are the factors that accelerate the onset of dementia and what are the factors that protect against it or underlie the resilience for dementia in people with aging, with Down syndrome, and especially with these genetic families, the Diane studies, for example, or API studies, where people have early onset gene mutations that lead to Alzheimer's.
Yeah, so it's pretty clear that not everybody gets Alzheimer's disease, and that there's the pathology is very different to people that do. So after 30 years of let's face it, no success in curing Alzheimer's disease, at least maybe we can prolong the phase at which people remain normal or slow the decline. So that'd be a big benefit for everybody if we can do that. Let's be normal longer.
Yeah, along with that thread, there's a big push for health span rather than lifespan, right? Like even if we're living the same amount of time, those last 30, 20, 10 years, if we can make them better, we're more capable, then I think that's a win for everyone. So one of the big threads in this conference and all the reading that I've been doing lately, of course, too, is inflammation. Now, we've covered inflammation in some previous podcasts in a basic sense, but inflammation in the brain is primarily, we think, mediated by microglia. I don't want to put words in anyone's mouth, so if I'm wrong, please let me know. Microglia being the brain's immune cell, and for me, it seems like they're a key target if we're looking early in the disease. They might be one of the first things to go wrong, or they might be trying to stop whatever's going wrong early in the disease. So it's really hard to understand whether they're the good guys or the bad guys. And I was wondering if we could get everyone's opinions on that.
Well, I think it's a bit of a stretch to say that microglia are the only cells that have an immune response. I mean, that is their job, and they are professional immune cells. But every cell in the body has an inflammatory response and its innate immune response. If sponges can do it, flies can do it, sea urchins can do it, any cell can have an immune response. So I wouldn't rule out immune responses in the other major cell types like neurons, astrocytes, right, oligodendrocytes too. That's just my bias, but I'm trying to be unbiased by having that bias against unbiased.
That's fair, yeah.
Okay, this is Leah again. Opinion of a pathologist. There are very classical ways that inflammation is defined in classical pathology, and these new discoveries in the brain is really changing everything. It's different paradigms, and we are still learning about it. Inflammation usually is a reaction of our body to protect us, to protect against infection, to help us to heal. So just to call inflammation something that's bad for you, maybe it's a little bit too strong. But what we are seeing from the growing evidence in this field is that what's probably going on in the brain that is contributing to dementia is some part of this inflammatory process probably going a little bit wrong or to the wrong direction. And the question is if you will be able to identify what is this wrong direction and bring it back to the normal status. And I think this will be interesting to see how it develops.
Well, it's always been a double-edged sword, right? I mean, you can have too little of immune response and you get infection and you die of infectious disease. You have too much, you have autoimmune problems. So the trick is to be in the right spot.
Yep. And this is Cindy. I completely agree. And if we're allowed to kind of go out on a limb a little bit, what I would say is that according to very early pathologic data, microglia, you were asking, Connor, specifically about microglia. So first of all, there are many more astrocytes in the brain. They don't get enough attention, but I think that's changing now. But microglia are very important. And microglia typically are not associated with the very earliest deposits of beta amyloid in the brain. So the diffuse deposits, for example, typically do not have microglia associated with them. My personal opinion is that people walking around with a head full of amyloid, the reason that they're allowed to do that or able to do that without having dementia is possibly because they don't have a really strong immune response to that amyloid. And by a strong immune response, I mean activated microglia attacking the plaque, also trying to get rid of it, and also reactive astrocytes. And so it may be that there's this whole sequence of events that occur, and if somebody's able to stave off that immune response for a little while, they may be protecting the downstream effects. So I think that the inflammation or the immune response is actually much closer to the neurodegeneration, and that may be a more common mechanism, not just specific for Alzheimer's, but for other forms of neurodegeneration as well.
Why don't people study astrocytes like they perhaps should. Is it just because they're very difficult? I mean, I know some of the markers aren't necessarily where they are. Like IBA1 is a great marker for microglia, and we can really lock them down. But I don't see so many reliable ones for astrocytes. Anyone have thoughts on why?
No, it's just like Rodney Dangerfield. They don't get any respect.
But you're right. It's more difficult to study astrocytes. They are plenty. The neurons also are plenty, but astrocytes, they come in many flavors and they change in reaction to injury or something. Neurons change less, so they are more stable in this sense. And in terms of markers, even for microglia, you mentioned IBA1, but there are probably 10 markers for microglia, maybe more, and we are still starting to learn what do they mean.
Right.
There is also, for instance, a macrophage that might cross the blood-brain barrier and they transform itself. It's something that it's half of a microglia. So, and they have different markers. And I think we're still, it's still incipient kind of study, but I'm seeing a lot of labs turning to astrocytes right now and microglia, especially if the single cell analysis, because it's easier kind to understand what cells we are looking at. So I think this field will explode, but I also feel it will be very, very complicated to interpret all the results. Yeah.
As it always is. That's good to hear, though. Absolutely. So all of this makes me think of the debate between the amyloid camp and the tau camp that I want to say used to exist. I know some people are still on one side or the other. In my mind, it's starting to converge towards the amyloid cascade hypothesis, and I imagine some of you might have some strong feelings on that as well. So the amyloid cascade hypothesis, for the listeners, is the idea that amyloid beta deposition or oligomerization, circulating amyloid beta even, might induce conditions of inflammation or hyperactivity within neurons that would then result in tau phosphorylation, aggregation, and ultimately killing the neurons. But people have different understandings of the amyloid cascade hypothesis. So am I just wrong in saying all of those things?
Yeah.
No, I actually don't think you're wrong. I think that there's some misunderstanding or misrepresentation of the amyloid cascade hypothesis because people consistently say, well, cognitive decline correlates best with tau pathology or with neurodegeneration, synapse loss. And that's true. But that's like looking at this long cascade and you're saying, okay, we're starting here at A. I know I shouldn't be pointing here, but you're starting to the left and saying, here's, amyloid deposition, and nothing really is happening to the neurons until you get to, let's say, S or T. And at that point, you start to see changes in tau and or synapse loss. And then by Z, you've got neurodegeneration and cognitive decline. It's a process. None of this is happening overnight. People have to remember Alzheimer's disease is a very long chronic disease. It doesn't happen overnight. And so, there may be early stages that may include amyloid. The genetic evidence for amyloid to me is like the key component. It definitely is a key player. Whether it's the actual instigator, who knows? But it's a key player very early in the disease. But it by itself is not sufficient to necessarily cause the neurodegeneration. So you have to look at the big picture and look at the whole temporal sequence before you can say yes or no. So that's why I think both are important, amyloid and tau, but I think there are a lot of other steps in between and after tau that are really critical.
Well, I just said the argument's moot because they're both amyloids. So it's just a matter of what flavor of amyloid. You have to have both the beta amyloid and the tau amyloid to have Alzheimer's disease, but you also get synuclein amyloid sometimes and TDP43 amyloid. So it's an amyloid-related disease, so the amyloid wins. But
so amyloid, you mean structure, right? It's a specific
amyloid
type
is a particular type of structure that they both make. And the fundamental distinction is it's an intermolecularly hydrogen-bonded beta sheet. So every strand in the sheet is from a different polypeptide chain. And that's what holds the aggregate together.
So you had a really good illustration. Hopefully I can get the slides, actually, from the conference of the different folds of origami. So when proteins are folding, amyloid beta and tau are known proteins that will misfold, and then we later question the word misfolding. But these proteins take a shape in which they're normally not found in their functional state. And you said that basically a sheet of paper can adopt all of these conformations.
Well, I think that's a peculiar property of amyloids, and it seems to be common for probably all of them. At least it's been shown now for A beta, tau, and synuclein. And what's unusual about it is people generally think that if you have one sequence of a protein of amino acids, you give rise to one native structure. For amyloids, it's different. You have one sequence that forms a sheet, and then that sheet can fold into multiple different structures, just like an origami can be made. Different origami can be made from a single sheet of paper. And it looks like those different shapes are important because you can clone these different structures out of diseased brain, and you can show for both A beta amyloid and for prion disease that a different structure is related to a different disease phenotype. So for example, there's a phenotype of Alzheimer's disease that is rapidly progressing that's misdiagnosed as a Creutzfeldt-Jakob disease, which is a prion disease. And it turns out it has a different structure than the most common structure of the late onset Alzheimer's disease. So this phenotypic switching or plasticity is important for disease, but it's also a problem of how you target it. It causes some issues about how you diagnose it, whether or not there are different structures involved with different subtypes of disease, and whether you have to target those individually like you target cancer individually depending on what type it is.
I think it brings a very good question is if Alzheimer's disease is a one disease or different diseases that manifest as the end stage as beta plaques and neurofibrillary tangles. So again, making analogy of other organs, if we think in cirrhosis, in the liver, you can have many etiologies, and neuropathologically they will look very similar. So the question is, maybe the field should look a little bit more of understanding this familial Alzheimer's disease as probably having a slightly different mechanism that sporadic Alzheimer's disease that will manifest later on in life, that has a lot to do with ApoE4 risks, which the familial states don't have and manifest in a different way. And coming back to your question, if the amyloid cascade, if it's something that's still current or not, I think if we feel in the concept that the beta amyloid cascade, and now I say beta amyloid, not any amyloid, as the upstream mechanism leading to Alzheimer's disease, maybe probably there are many people that will not agree with this, especially for sporadic cases, but I think that everybody will agree that amyloid is important. When we look at tissue, again, post-mortem tissue, we usually detect tau changes before we detect beta amyloid changes. It might be a problem of detection because we are not detecting oligomer forms that might come earlier and might be toxic using our traditional neuropathological method. So I think this is still an open debate. But also we have the oligomeric tau forms that we are not looking at. And certainly we need a re-examination of how the disease progress in the brain and what are the characteristics using the most modern techniques. I think the field needs this a little bit, a redefinition of what exactly is Alzheimer's disease in humans.
Definitely. So you mentioned some important techniques there, right? Well, the first thing that comes to my mind is antibodies and using antibodies. Now, antibodies are, in theory, ideally, basically a custom fit tag for a various protein or a piece of a protein that you want to look for. You can even sometimes detect post-translational modifications, so little alterations on those proteins and these subtle differences. Now, Dr. Glabe, today you had a presentation which basically said, yes, some antibodies work in some cases, but we cannot trust them. And you're looking into figuring out exactly these small nuances between antibodies that are supposed to detect the same thing and trying to figure out why exactly they're binding to different parts or showing you a different pattern. Is that a fair assessment of what you were
What, you
talking
mean
about?
You can't trust an antibody? What are they going to lie to you?
Sorry,
I think if you listen to them, they'll tell you the truth.
So I'd love to know your thoughts and why you started down this path of trying to characterize all of these different amyloid beta and
it's really
tau
simple.
antibodies.
I've been making antibodies since I was a graduate student. In fact, I'm blood brothers with a rabbit because I cut my finger when I was trying to bleed it. So I've been making antibodies a long time. And when I first got into Alzheimer's disease, my entree to the disease was actually chemically synthesizing the amyloid A beta peptide. So I made a gram of it because it's not that hard to make a gram. And so when you got a gram of protein, what do you do with it? You make an antibody. And so I made an antibody against the peptide, and I noticed it has this peculiar property. It only stained or reacted with aggregated forms, and it didn't react with the monomer or the precursor protein that has the same sequence. And that was completely foreign to me. I'd never seen this before in an antibody. And I think it's really simple. It's because the immune system is geared to recognize things that are pathological, and it's the aggregates that are pathological and not the native protein. So this is very typical of amyloid antibodies, is they don't recognize a monomer. You have to really work to find one that recognizes a monomer reliably. So they're pathology-specific tools. But the other thing that's pretty crazy about them is that there's so many. I mean, it's a small peptide. It's 42 amino acids. And here you can have dozens of different antibodies. And the reason is, again, it folds differently. So depending on how it folds, it will display a different surface topology. And that different topological features give rise to different antibodies.
I just want to mention that that in addition to the antibody binding, the way that we use antibodies to detect proteins in the brain also depends very much on the fixation of the brain, the postmortem interval, and the processing of the brain. And so we have a special collection of tissues where we've taken the tissue at autopsy and just fixed it for 1 to 2 hours, which is very atypical. Usually pathology departments will fix the tissue for at least a week, but sometimes even up to a month or so too, before they start sectioning the brain and looking at it. And that can block, you get these cross-linking of formaldehyde, and so that can block the binding of the antibody to the protein in the tissue. And I think that initially led to a lot of misconceptions about which pathologies were occurring in the brain. And I just want to go back to something that you said, Leah, which is, at least in Down syndrome brain, the amyloid pathology that we've seen, especially the diffuse A beta, was very, very strongly detected with antibodies before we saw any tau pathology. However, we never really looked at the brain stem. We looked at cortex, hippocampus, and cerebellum. We never looked at the brain stem. So, I think now that we have newer antibodies, better techniques for looking at the brain and really understanding the temporal pathology, I think it's really important to go back and really map it out in the brain and try to figure out the exact pathology.
I'm an end user of antibodies, have been doing this for years, but I find amazing how the field improves. Not only the antibody itself, but the techniques that have been invented to recover the tissue and being able to use the antibody, what we call antigenal retrieval. Now we put tissue in the autoclave to very hot temperatures, and then we make any kind of antibody work, so to say. It's amazing how things change. We amplify things. We have a lot of tools that we didn't have before. And again, this is why I feel that we should do a systematic re-examination of how this disease manifests in the brain in terms of pathological changes, because this is so fundamental to gear our research with mechanism and cells and so on, and our concepts. And I think we are ready for doing that with these antibodies that you, for instance, produce. Again, I'm just an end user.
Right.
It's true, but we got to be careful about antigen retrieval because it is like black magic.
Right.
And at the bottom, the bottom line is what it means. Antigen retrieval is I like what I see, and I didn't like it before.
And an autoclave, for those who don't know, is basically like a huge super powerful pressure cooker. Superheats the sample with super high pressure. It's mostly designed to just sterilize stuff, to kill anything living existing there. And
it's like scrambling eggs.
Which that's actually really interesting because Dr. Grinberg, you're 3D printing patient skulls, people who donate their brains for research post-mortem. You're 3D printing their skulls to then house their brains to keep it more stable so that you can stabilize the brain slices that come out very thin and protect them from damage, which would stop you from being able to analyze them. And you can look at these really delicate regions like the brainstem. So, I mean, can you talk a little bit about like who came up with that idea and how all that works in sequence?
Sure. So we developed this idea because, as I said, it's very important in my research program that we can bring the knowledge we can see in post-mortem tissue to living patients. And we feel the neuroimaging like MR and now PET scans and all these modalities really can be a window to the brain. It's just that they have still a much poorer resolution that, a microscope. And I don't think it will change so much. So we came up with this idea that if we would be able to correlate or overlap exactly point-to-point neuroimaging with the brain histological reconstruction of the same patient, we will be able to learn things that we wouldn't see with neuroimaging and transfer this knowledge for the next patient that won't come to autopsy. And one of the biggest problems we had is by removing the brain from the skull, it always gets deformed. So the fresh brain is like a gelatin, and we have to fix it somehow. And there's always either we'll put more water inside or remove more water in a way that we cannot predict. So at the end of the day, it was very difficult to do this overlapping. So we came with this idea, and if we could fix the brain in the same kind of compartment that the brain came from. Oh, I had a fellow that came from Brazil, a neurosurgeon. He was called Eduardo Alho. And he said, how about if we print, or he said it would be so nice if he could print the skull. And I found it was a great idea. And then we were thinking, how can we do it? Do we have to make a mold? We cannot make a mold. It was impossible to do it until we started to get 3D printers in the campus, and they needed beta testers. So it took, so we volunteered to be a beta tester. What took longer for this is to learn how to bring, this CT skull images and transform this in a 3D model that we could print. And we could do it with the help of another group from the University of São Paulo in Brazil, in which they model all organs in 3D for teaching students in universities that don't have autopsy. So they help us to fix our problem with the skull. And now we do for all the patients that undergo this kind of research, and no deformation anymore, much easier to overlap.
Wow, that's amazing. So each brain comes in its own custom display case? Like
it does.
Jewel?
It does. It's custom made.
Cool. Very cool.
We'll definitely have to put a picture of that in the show notes so people can really just understand how that's all working together. And one of the reasons you wanted to do these brain scans, right, is to look for early markers of Alzheimer's disease, give people an early warning signal. So first off, I have to ask, do you think everyone's going to want to know super early on if they're going to get the disease? And how would you handle those diagnosis criteria? I know we're not clinicians here, but maybe an easier question, would you want to know? And
I'd only want to know if it could be treated.
I think there is a big difference between what we do for research and what we do in the clinical setting. Mm-hmm. So there are many people who understand the value of going to research and help us researchers to understand better about the disease. At this time point, we don't have a prevention for Alzheimer's disease. We don't have a cure for Alzheimer's disease. So I would not recommend someone just to go and check if they have markers of Alzheimer's disease, because I think they will be very anxious that there is nothing really to do. We cannot even tell them if they have these markers that they will develop dementia or not. So we are not yet there.
Yeah, and even if they don't, it doesn't mean they're not going to get it next month.
Right. But for research, we do have all these cohorts of healthy aging people that come to our centers, and they come back every year, and we look for these markers and we follow up what happened to them with this hope that we can understand better about the disease and one day bring this understanding for the population. So in this sense, I think the research is important, and it's important to detect all this. But I have to say that many of our research participants, they don't want to know if we found the markers or not. They are okay coming for research, they come every year, but they don't want to hear about it, which is interesting.
But I think, if I remember correctly, Gil Rabinovich has done this study looking at people that have had amyloid PET scans and whether or not receiving that information, that they had a positive PET scan, was useful or helpful to them in planning ahead and for Medicare purposes, all of that. And my understanding is that overall, the outcome of the study thus far is that people do want to know. The people in his study did feel that it was helpful to learn their diagnosis, and that it did help them and their doctors sort of customize their plan for care.
And the age of personalized medicine is upon us, I think. So all of this information, if we can compile it and really process it, is going to go a long way. That also brings me to one of the major risk factors we've discussed at length is ApoE4. Now people are doing genetic tests for this. 23andMe is a big example. Have any of you done genetic tests for the variant?
I did, and I was happy to hear I don't have a ApoE4, but I was very nervous about it. And first I wanted to do this test because of ancestry. I was very curious. And it just came in the package, and I was debating if I would like to see it or not. My grandmother had Alzheimer's disease. She developed at the age of 70, which is not considered early onset, but relatively young. And I think it was one of my biggest fears to have a ApoE4. So I have to say that now I feel much better, but probably if I would have a ApoE4 or 2, probably I would have regretted very much. So it was a kind of a lottery. I don't recommend.
Really tense moment, I'm sure, when you open the results.
Yeah, I didn't do it. I don't know why. I mean, I could, but it's just not something that I was interested in. Still not interested. Yeah. Maybe I'll find out I have some relatives somewhere that are unsavory.
And then they'll want to come visit. Yeah, that would be a problem.
Right. Yeah.
You can do that without ApoE4.
Well, yeah, there's all kinds of markers. You never know.
So you're all involved with BrightFocus in some way, and I really appreciate all of you coming to this conference because it's super geared towards me, the student, and postdocs, developing scientists. It's given me such a great platform from which to pose new questions in my research, from which to adjust my parameters in analysis, to really think about all of these things working together in a common system, and then also just the extent of collaboration between so many Alzheimer's disease labs. If you could each tell me, I guess, how you got started with the organization. Now, Dr. Grinberg and Dr. Glabe, I know you both have grants, research grants, on some of the subjects that we've just discussed. How did you hear about it, and what are you looking forward to moving forward in the organization?
Well, I don't know. I mean, I've been around this foundation forever. Actually, I got my first grant to work on Alzheimer's disease from the precursor of the BrightFocus Foundation, which used to be called the American Health Assistance Foundation. And that was some 25 years ago. And after I got that grant, I've been working for them, reviewing grants, doing things like that for a long time before they had this workshop. So coming here is just like Old Home Week. And just to show I didn't abuse my friendship with all the people at the foundation, I never got another grant for 25 years until last year. So
no conflicts of interest.
Well, I mean, everybody's conflicted to some extent, but I didn't work on the grant review panel. You can't when you're applying for a grant. But the important thing is I didn't apply for a grant during that time. I could have, but I didn't.
I got involved with the foundation for the first time some years ago when one of the organizers of this meeting, Harry Steinbush, invited me to talk. And I was really amazed by how important it was to put together so many trainees in the room with very high quality faculty. I mean, the faculty here is stellar, and I feel kind of humble to be, part of this a little bit, not the stellar, but to be part of the faculty. And then I applied for a grant. And actually, it's a grant that we use these 3D skulls to try to detect very early changes in Alzheimer's disease. And then he invited me again this year, and it was a pleasure to come. I wish there would be more of those opportunities for trainees. I think it makes the difference. I see many people who start in one of these trainings and really set sail in their career in this area with very good network and knowledge. So super, super important. And one of the first trainings I went when I was a PhD student also has been organized by Harry. It was in something else. But I met many people there that I collaborate until today. It was super important. So I feel happy to come, and I think it's time well spent.
And I have been involved with BrightFocus for many, many, many years. And like Charlie, I have served on the Grant Review Council for 11 years now, I think. And as such, I'm not allowed to apply for grants, but I did have a postdoc who had a fellowship a couple of years ago, which was very helpful for her in her career. But what I've really enjoyed a lot is being a co-organizer with Harry and Frank. It's been a wonderful experience. I don't even remember when the first, my very first time coming to the workshop, but before it was specifically BrightFocus, it was AHAF and ISOA. And I went to a meeting that Harry organized in the Netherlands and gave a talk there. And then I think I've been part of the BrightFocus Foundation either as a speaker or over the last 3 meetings as a co-organizer. And I have to say, I love it. I truly think it's one of the best, if not the best, trainee workshops in Alzheimer's research. I've made tremendous friends and friendships through this meeting, both with the faculty and established new collaborations, but especially with the students. And I've had 3 different students come to my lab for different periods of time to do research. And what I really like is that we stay in touch. This is not like, you come for 3 days and then you leave and you don't talk to anybody again. This is really a way to establish a path forward and to keep up the network and keep up your friendships and your collaborations. And it's really a great opportunity, both for the students and the faculty.
Yeah, and I've seen that already. I'm interested to know, you've watched the BrightFocus Foundation and then the AD Fast Track Conference grow over the years. What's changed and where do you think we're headed next?
Well, I think it's grown a lot bigger than it used to be. It's the science, I think, has evolved to be much more sophisticated. I mean, science in general, technology has grown. We continue to get incredible speakers. We're extremely lucky and very grateful that we get such great faculty who agrees to come to this meeting. But I think part of that is that the reputation for this workshop is really growing. I think people respect it and want to participate, and hopefully we're making it engaging and fun for the faculty as well as the students. I also think that it's great having it right before SFN, which makes it convenient because a lot of people are going to the meeting. So this is like an add-on. It's great to have it right ahead of that. I believe that the plan now is to maybe go every year rather than every other year. It was every other year, but I think it speaks to its popularity and its success that now people want to have it every year. So hopefully that'll be the next step is to hold this every year.
Annual meeting. Yeah, that sounds great. Along the lines of growth and progress, what are each of you most excited about in the field of Alzheimer's disease or general research? I'm love to know your thoughts on the trends.
I'm really excited by the fact that the NIH got over $800 million more in research. So it's going to really stimulate a lot more work. And if we don't have a treatment in 10 years, I'll take the blame. It's on me.
You heard it here first, yeah.
I see much more initiatives to cross-pollinize and understanding from different researchers from different fields. There are more consortium form. And I have been exposed to some kind of science in the field of Alzheimer's disease that would never be in a traditional setting, that I will be in my lab, going to the neuropath meeting or going to the neuropath session, in whatever conference. And this is very exciting for me because I think with these different contributions, we can see the disease in a different way and probably advance in maybe right direction. I don't know. I mean, right direction is when we get the treatment. I don't know when we get there, but certainly we had some bumps in the way so far.
Yep, I agree. And just to add on to that, I think what's exciting for me is that people are really starting to look at Alzheimer's disease in the big picture, not just looking at the brain, but looking at the whole body and looking at the microbiome and the peripheral immune system and cardiovascular impact on Alzheimer's disease. And so to me, that's really exciting that we're looking at the whole body, the whole person. And I think that will also help us get to a treatment, or maybe not a single treatment, but customized treatment, precision medicine, or combination treatments that may be much more effective than what's been tested so far. I think the other big thing that kind of blows my mind is big data and all of the omics and all of the research that's coming out of that. It's so powerful, and I can't believe that that would not lead somewhere really important. So I think that the horizon is very bright, and that we have optimism at this point that we will learn from all of this big data and from all of these collaborative studies, a consortium. There's a lot more sharing of data now than I think there ever has been in the history of this field. And that's all a good thing.
The other thing that's exciting is we got the first disease-modifying treatments reported ever. So we got 2 treatments. They're both monoclonal antibodies against amyloid A beta, and they slow the decline, the cognitive deficits. So it indicates that A beta might be a good target, and we just need better treatments. But at least we can see something on the horizon where we didn't see anything before.
Yeah, it's super promising. Would you say the jury's still out in terms of amyloid versus tau? Because there's an anti-tau antibody therapy coming down the pipeline too. Are you hopeful, just as hopeful for those antibody therapy results?
Yeah, I'm ecumenical. They're both amyloid. There's just the flavors, kind of like Baskin Robbins.
Right.
Not quite 31 flavors, but they're all good.
And I would just say, again, it's not one size fits all. So it could very well depend on the stage of the disease that you're treating. So if somebody's already got a head full of amyloid and they've already started the downstream changes, removing amyloid at that point may not be effective. All indications show that it's not going to be effective. And that might be a point where you want to look at tau as a target. But if you're looking at primary prevention before anybody even starts depositing amyloid, or secondary prevention when they have amyloid but they don't have neurodegeneration, they don't have cognitive decline, beta amyloid, might be still a viable target.
One thing that was exciting for us lately, we published a paper in which we could show that people that pass away, and the only thing they had in their brains was some tau deposition in subcortical nuclei, not really in the cortex, and they didn't have amyloid deposits, at least yet, compared to people at the same age that didn't have anything in the brain. They had a higher risk of showing some symptoms that usually we don't associate with Alzheimer's disease pathology, like depression and anxiety and agitation and sleep disorders. We know that these symptoms, they are very commonly seen in people that develop Alzheimer's disease, and we always consider, is this a risk factor or not? And we could show this. So what does it mean? So for me, at least, if you forget about Alzheimer's disease, memory loss, and you think about the aging population, and you see the most common problems that people complain when they go to the doctor is that they don't sleep well, they don't feel as excited anymore, or they are agitated. This is very common things. We don't really have a good treatment for this. We treat with symptomatic treatments that have, some effect in some people, but mostly it doesn't last for so long. So the question for us looking at this result is it possible that maybe at least for part of this population that are having these complaints, they have it because of Alzheimer's pathology in the brain? So we will be able to use these drugs that maybe are now showing disease-modified effects that we think, let's prevent memory loss, also to prevent or to improve depression and sleep changes and agitation in this population. This will be huge. I mean, it has a reach that's even much bigger than the memory loss that's already devastating and very prevalent in Alzheimer's disease, but touches some other problems that we see during aging that can be decrease the quality of life of people.
Yeah, that's great, and brings me to one of my favorite questions to ask just about everyone to come onto the podcast. How has something or the whole process of learning throughout your research, how has that changed the way you either look at life or live your life? So one example for me is, Dr. Grinberg, your research has shown that the locus coeruleus is one of the first places to get hit with Alzheimer's disease pathology. So it seems to be vulnerable. And one of the important things about the locus coeruleus is that it releases noradrenaline throughout the brain. And that's super helpful in terms of just regulating normal function, and may have some links to sleep and depression, like you just kind of mentioned. Now, I've heard that cold showers induce noradrenaline release. I don't know if that's necessarily true, but I started trying it out, and it's a shock at first, I have to admit, but it does feel really good afterwards. Has there been anything like that for any of you in your research path?
Yeah, I'm addicted to discovery. There's no bigger rush in life than gaining insight when you're looking at a new piece of data. You get this feeling, you go, wow. That's why I do it. I'm really addicted to it. I got to get my fix. You never know when you're going to get it, though. It's not like you can go out and buy it.
Yep, that's true. I would say that all of the research about exercise has stimulated me. I have my Fitbit on, and I'm like addicted to at least 10,000 steps a day. But the data looks really convincing that it really is helpful, and it's not that difficult to do. The biggest problem is, we're all sitting around writing our grants and writing our manuscripts and, trying to find the time to go out and grab a walk after the afternoon session before dinner. That kind of thing is difficult, but it's really important. And I find that it keeps me balanced way more than when I don't go walking. Like, I actually miss it now. If I don't go for a walk every day, I really miss it.
Oh, but that's easy for me. I walk to work every day, about 3 miles round trip, so uphill and downhill.
That's great.
Yeah, I bike to work, which is also good. And I also, I believe this, all this evidence of exercise, not only for the brain, but for aging in general. It's so important. I make it like a commitment every week that I will do it. It's not even more choice, it's like a part of life. And I enjoy, I need my doses of endorphin. I don't eat chocolate, so I think exercise kind of try to replace it. It's good.
I used to ride my bike too. It's wonderful, but I got tired of getting hit by cars and ending up in the hospital, so really? I decided to walk.
How many times does that
happen? But I think
it's
something that it's something that everybody can do, or most people can do some form of exercise, and it's easy. It doesn't cost anything. You can go for a walk, ride your bike, whatever. But it's really, I think it's one of those things that we can all do. We don't have to go take memory tests. Just go and do your exercise.
It's free.
It's free. Yeah.
Yeah, and Dr. James Hicks had a great presentation on that earlier today. I'm hoping to get him on. He'll probably have to come in virtually because he had to leave early, but super convincing evidence that it supports not only, like you said, normal aging, but as well in the pathological
You can talk to Mike Yassa about it too.
Mike Yassa, okay.
Yeah, he's the senior author on it.
Oh, right. Yeah.
Well, and it's also, I mean, it's really good for your cardiovascular system. And there's so much evidence now that the brain and the heart and the cardiovascular system are all connected. I mean, we're not
They got pipes.
just looking at the brain. We have to look at the whole body.
And we also have to remember we are talking a lot about Alzheimer's disease pathology as a driver of dementia, but we know that it's very rare that someone just have one driver of dementia in the brain. And we talk about EDP, we talk about synuclein, but vascular changes in the brain, they are very important, and they do accelerate this transition between being normal with Alzheimer pathology and having Alzheimer pathology and not being normal. We saw by the NAN studies and some other studies, and they are prevalent. And to prevent this are the same things that we use to prevent heart disease. We have to keep, our blood pressure, especially before midlife, controlled, and cholesterol and exercise. And I mean, all the same recipe for the heart. I think it will help the brain for sure.
So the vascular amyloid is just another flavor of amyloid. It's like it's number 8. So we haven't got to 31 yet, but we're on our way.
Thank you all so much. Again, this conference in general, this conversation, super illuminating for me. And I'm going to have to process all this and write it all in my notes. Luckily, I have some of it recorded for posterity and for myself. Is there anything else you'd like to say to our listeners, anyone who would come across this podcast?
Exercise.
Exercise. And I'd also like to thank Connor. I'm very impressed. I think you're doing a great job. Thank you for doing this.
Thanks.
Thank you. Pleasure to be here.
That's going to be it here for us at the Straight From Scientist podcast. Thank you so much for listening, and especially if you provided feedback or are a sponsor of the podcast. If you'd like to become a supporter, you can go to patreon.com/SFS podcast.